Right now, what I'm going to tell you, we all are wrong. This is not the correct answer, because 3 marks, you can understand just calculate the mean value. We don't get 3 marks, maximum 1 or 2. So then I will show you the technique. What is the correct way of doing this? Okay, so let's look at the data again. Now the student repeat this experiment who done in tube 1, first tube, first, second, third, and fourth. And if we go back here, so why they wrote here, repeat the method for 3 more times. So here they say 3 more times. Okay, repeat the methods for 3 more times. It doesn't matter. And here we have a data for 4 times. So this is a data collected for 4 times. Now the problem is, you can notice the value, this value, 1.89. 1.89. It's not working. One 1.89. Now look carefully all the values. 0.55, 1.89, 0.54, 0.61. Right. Now, can anyone, if you know this, give a name for this value? If we have a group of values, and if you look this value, it doesn't fit to the pattern. So 0.55 and 0.61. This value does not fit to the pattern. So what is the name given if you have values like this in a collection of data? So we call anomalous value. These values are called anomalous value. Anomalous value. This value is called anomalous value. The meaning is the value which does not fit to the other values. So you can write your notebook, what is the meaning of anomalous value? So write in your notebooks, anomalous value. And then you write the value which does not fit to the other values. Example, you can write this value 0.55, 1.89, 0.54, 0.61. And then you can round this 1.89, and then you can write this value is anomalous value. So in your notebook, you can write this information, anomalous value, and define anomalous value and give example using these numbers: 0.55, 1.89, 0.54, 0.61, and round 1.89. Then you write this is the anomalous value. Then you can also write we do not add anomalous value when calculating mean value. We do not add anomalous value when calculating mean. So you have to remove this value and you have to add only 0.55, 0.54, 0.61. Now can you calculate the correct mean value after you copy this note? So that's again very important information. You should not forget this information, the value which does not fit to the other values of the group. Example is given here, and we do not add anomalous value when calculating mean. Now you can use these three values to calculate mean value. So what you have to do, add these values, all values, and divide by 3. So 1.70 divided by 3 should be 0.6333, at least 6.63. So I do it again. I did manual calculation. 5 plus 5 is 10, 17, 1.70. So divide by 3. So others, can you please calculate what is the new value now? Add all the numbers, divide by 3. Yes, I think that's correct answer. So this one I should make manual corrections. 0.63 17, 3 times 5. 3 times 5 is 15, 20 left. 6, 6, 6. So then round it up to 2 decimal. This 6 is higher than 5. Then I have to make this 7. 0.57. The correct answer is 0.57 now. So now you can write correct answer here. 0.57. 0.57. Okay, I think this is something you learned today new. And there are many techniques you have. It's not just like biology is, you just learn and memorize something and write it. We have many applications. This is the one important thing. Now, today you learn how to remove the anomalous value and calculate the mean value. So the value is completely different. For example, earlier we got 90, now we got 57.57. So this value is wrong. It doesn't fit into the mean value. This is the mean value now. This is the correct mean value. So this kind of a question you are always getting in the exam, and you have to remember, remove anomalous value and calculate the mean. Okay, I am still giving the chance for you. Can you find answer for this and send it to me for the second question number 2, Roman number 2. What substances are produced when gelatin protein is digested? So please send answer for this question. Netuli and Litun. Yes, I think you send the correct answer. Right, I've given a hint for the others to find the correct answer. Gelatin is a protein. So now if you write protein here, break down by enzyme, protease. This is your knowledge from digestion. What does it make? Protein break into its building blocks. So protein going to make its building block. What is the building block of protein? This is not good sign. You have to study. That's called amino acids. Amino acids make protein. So answer is amino acids. Amino acid make the proteins. So keep on studying digestion. It's very important. What is enzyme? Where does it happen? And what is the end product? Starch, amylase, maltose, maltase, glucose. So enzyme, substrate, and product. So you have to remember what is substrate, what is enzyme, what is product in digestion. Okay, now here the student repeat the experiment with different pH buffer. This time student use different pH buffer. They change the pH. So you can see pH 3, pH 5, pH 7, pH 9, pH 11. The student changed the pH. This is the result. 0.38, 0.9 is the highest, 0.32, and again 0.51, 0.33, and 0.01. So then I just draw like this to show the highest digestion happen at pH 5. Then low pH less, higher pH also less. Give two variables the student should control. Okay, now you have to write in your notebook. So you have to write in your notebook this important thing. So what is important thing? What is you have to write what is independent variable? The independent variable is what we change. So you write in a notebook, what is the meaning of independent variable? It is what we change in the experiment. Dependent variable, what we measure in the experiment. what we measure in the experiment is dependent variable. This is very important to remember, independent and dependent variable. Independent variable and dependent variable. Independent variable is what we changed. Dependent variable is what we measure. And you can see in this question, control variable. Variable that should be controlled. So there's another one, control variable. Control variable is what we keep constant in the experiment. What we keep constant in the experiment. Right now, I give a small exercise. If you copy this one into a notebook, I will give a chance to identify independent variable of that experiment. So you can copy this first, and then we'll see whether you can still identify independent dependent variable from the result. Right now, looking at the result, can you try to find what is independent variable? From this experiment, what is independent variable? What is independent variable of this experiment? The independent variable is what we changed. Okay. Uh, it seems none of them are very good in this experiment type of questions. I think so far maybe you have learned the theories. Maybe you haven't practiced any questions for next May June exam. We have few months: September, October, November, December, January, February, March, April. So I think 7 months left for your exam. Just 7 months left for your exam. Uh, it seems that you had to work more. Right. I will show you again what is independent variable is, what we change. So you cannot identify what the student change here. Change the pH. So pH is the independent variable. pH is the independent variable. So independent variable is pH. So what is the dependent variable? What student measured? Student measured the volume of gelatin digested. This is dependent variable. Volume of gelatin digested. Now, when we finish independent and dependent variable, the student has to keep other variable constant, control variable. So you can't use this pH, you can't use because it is an independent variable. So can you name one control variable? What are the other things the student should keep constant? So again, the same experiment, only change the pH. So he used this setup: pineapple juice pH buffer, gelatin, protein, everything he used, but he used different pH. So what are the other things student keep controlled? At least one control variable. pH is changing. You can't use pH. The student should keep other variable controlled. Okay, example temperature. Then the volume. So, for example, student use always 5 cm cube, 1 cm cube, and 1 cm cube of pineapple juice. Temperature 37. So these are all always constant. Otherwise, it is not a fair experiment. Volumes, volume of pineapple juice, and the temperature. Volume of pineapple juice and the temperature. Okay, I will check your theory knowledge before we finish that question. So what is the test we use for protein? What is the test we use to check proteins? So maybe I think all of can answer. What is the test we use to test for proteins? Okay, I think you have to really study. This is a very basic thing. I think if I remember, we have to learn from class 8. We are learning this from class 8, 9, 10, all the levels, the protein test. I feel you need to study a lot because some did not answer, some did not answer correctly. The test is bio-red test. If you don't know this, and 7 months is not enough for you to study because we have an advanced question like this. For example, this kind of a question is your exam question. Explain the effect of changing pH on the mean volume of gelatin digested. So, yes, I know that only one send the answer, others are not correct. Yeah, so I suggest now, I got some idea about your levels. You have to really study the basic things again. So 1 year is over. That means you finish your syllabus. Now it is no time to finish syllabus. Because if you still waiting, the syllabus is not cover, I am going to study like that, then papers are quite advanced. You are, I feel still the practical question like this, you have never actually practiced this kind of a questions. And basic theory question, also you are away from the theories. It is right time now to study the theory. Then only we can get good mark for the exam. Okay, my message is after the first lesson, you need to start again to study from your theory because no one can finish theory in 7 months. If you finish in theory 7 months, no time for the papers. So our journey is first is the theory finish, second is revision and papers. So please study your theory, then we can handle question much better before because it's a little bit of not up to date with your theories. Okay, I'm going to stop here. See you next time.
Bye, sir. Have a nice day.
Bye. Thank you. Bye.
Night.
Bye.
Bye.